Good morning, dear students. Today we are going to start our section with DNA binding proteins. First point, which are the different types of proteins that bind DNA? They include histone proteins and non-histone proteins. Among the non-histone proteins, so many other different classes come there. Regulatory proteins, structural proteins, and motor proteins. We are going to learn about each one separately. Histones, I think it is very familiar to all because you have learned about this histones in your UG classes. And what about the nature of these histones? They are basic proteins. Basic means what? They are positively charged ones. And their molecular weight is between 11,000 to 20,000 Dalton. And also as it is basic, it is positively charged. Why it is positively charged? Because of the abundance of positively charged amino acids like arginine and lysine. Because of the presence of these two positively charged amino acids, these amino acids are abundantly present in histones. Because of this fact, these histones are positively charged. That's why it is basic also. Next, types of histones. There are 5 different types exist. They are H1, H2A, H2B, H3 and H4. This is a picture of histone protein. And what are its characteristics? It has a central non-polar domain, forms a globular structure, and an N-terminal end is there and a C-terminal end. And this N-terminal and the C-terminal end carries most of the basic amino acids. Then coming to most specifically to C-terminal end, what is its role? It makes this histone DNA and also histone to histone interactions possible. Which all interactions it is making possible? C-terminal end. First one, it makes that interaction between histone and DNA. At the same time, histone to histone interactions. Now, what is the role of N-terminal and specifically? They are the targets of post-translational. Post-translation means after the translation. Some modifications need to underline, and that modifications are happening on the N-terminal end. Okay. Then nucleosome core particle. How it is forming? 2 copies each of H2A, H2B, H3 and H4. They aggregate and form the core. It is called histone octamer. And wrapping around this octamer, we can see here, 147 base pairs of DNA is winding or wrapping around this octamer, and that structure is called the nucleosome core particle. Clear? That also you have learned in your UG classes. Next, linker histone. Because up to here we did talk about H1 histone. So here we are going to understand what's the role, what's the function, purpose of H1 histone protein. It is seen here between at the point where the DNA enters the histone octamer and at the point where that DNA leave the octamer. At both these sides, the H1 attached to this DNA and making this wrapping firm. Wrapping of this DNA around the histone octamer firm and helps to keep it in position. This explaining here. It binds nucleosome at the beginning end and the end site of DNA and thus helps to keep DNA in position. That's why it is called a linker histone. And we already know that the histones are positively charged. As they are positively charged, it can very easily link with the negatively charged phosphates of DNA. Negatively charged phosphates are seen in the backbone of DNA. Okay. Then we are going to understand about some modifications that is happening to histones and what the role of that modification. What is the significance of that modifications? Histone acetylation. If acetyl groups are added to some histone specific position, what will happen? What is the significance? Which enzyme is doing that acetylation? Histone acetyl transferases, the enzyme that is doing that modification. What's the effect of that histone acetylation, adding of acetyl group to histone? By adding acetyl group to histone, it reduce histone's positive charge. As the positive charge diminish, then it lose the strength of interaction with the DNA. That's what. As it have more positive charge, then only it can strongly bind with the DNA. As the positive charge reduce, that bonding, the strength of that bonding with the DNA reduces. Okay. That is the effect of acetylation. Then deacetylation is the vice versa of this effect. Which enzyme is carrying out this deacetylation, removing of acetyl group from histone? Which enzyme? Histone deacetylases. By the removal of acetyl group from histone, its positivity increases and thus by strengthens the interaction with the DNA. And one more point we need to understand along with this, because as the interaction with the DNA increases, it is limiting or preventing transcription. We know that for transcription, need to unwind the DNA to extend the DNA fully in a free in an extend form. Then only transcription will go very smoothly. So whenever that interaction between the DNA and histone is strong, that means transcription is not possible there. Transcription is difficult at those regions. Wherever the histone DNA bonding that interaction is weak, that means the DNA can unbind and thereby promote transcription. Then histone methylation. Histone methylation is done by the enzyme histone methyl transferase, and the effect of that methylation is that by methylation of histone, that interaction get weakens. As the interaction weakens, transcription can start very easily. Then histone demethylation is the agonist of this one. Demethylation means removal of methyl group from the histone. As methyl group is removed, the histone DNA interaction become more strong and it will prevent further transcription. That means it silences the DNA transcription. Then histone phosphorylation. Histone phosphorylation means it indicates a mark of or signal one DNA damage or DNA repair. And histone ubiquitination. Ubiquitination means that indicates that the chromatin structure can alter. Alter means it can unwind and is possible to give some access to enzymes that is involving in transcription. That is ubiquitination. Histone ubiquitination also promote transcription. Next, another type of proteins that is binding DNA. They are the function of these proteins are to regulate the transcription of DNA. So, regulatory function, these proteins, they are called the transcription factors. What is this transcriptional factors? They are the proteins that is controlling or regulating the transcription. Which are the different types of transcription factors? General transcription factors are there. Transcriptional activities are there. Transcriptional repressors are there. General transcription factors. What is that? They bind to core promoter region here. This promoter region is way away, means upstream at the start point of the G. Promoter. At this site, this general transcription factors are coming and bind, and this binding permit the enzyme that is involved, or the enzyme that is carrying out transcription, become able to come and join the specified position. So that is the role of general transcription factors, to help transcription or to initiate transcription by interacting with the RNA polymerases. RNA polymerases is the enzymes that is doing transcription. For that enzymes to bind and initiate transcription, it need assistance or that prompting from the transcriptional factors. Next is transcription activities. Transcription activities means, and these are another class of proteins that activate or stimulate the transcription of adjacent gene. Adjacent gene that is near to the place where these proteins, this activities bind. So suppose this is the gene and this is the transcriptional factor it bind, and then it can activate the transcription of its adjacent gene. That is transcription activities. Then transcription repressors means as it binds with a position near to a gene, it suppress the transcription of its adjacent gene. Okay. Next, regarding the regulatory sites in gene. Regulatory sites are the sites for the transcription factor to bind in the gene. If we take the case of a particular single type or a specific type of transcription factor, different gene have the same regulatory site for binding with a particular single type of transcription factor. Or other words, the same transcription factor can have regulatory site in different types of genes. And the other condition is that sometimes a single gene have regulatory site for binding with different types of transcriptional factors. Then what about the proportion of genes according for the transcription factors? In our genome, a genome of one cell, about 5 to 10 percentage of the genes are for producing transcriptional factors, that type of proteins. About 5 to 10 percentage of the genes of our genome is responsible for generating the transcriptional factors. Then regarding the structure of transcription factors. For the transcription factors, two domains are present. A DNA binding domain is there, and also an activation or regulatory domain is there. Two domain, DNA binding domain. What is the role of this DNA binding domain? Using this domain, it binds certain specific sequence of base pair in DNA. Then what is the role of this activation or regulatory domain? As the name implies, it regulates the transcription by interacting with some other proteins. That is the two domains. We show the image of that transcription factor. This is a transcription factor's image. The DNA binding domain is there and a regulatory domain is here. Dimer formation. In most of the occasions, they form dimer. Dimer means there's one transcription factor joined with another transcription factor of same type or different type, which will enhance its function. Next is motor proteins. Motor proteins are certain types of proteins that binds on nucleic acids. They include polymerases are there. Polymerases means they are those enzymes that is carrying out transcription. Then topoisomerase transcription is there, DNA replication is there. So many functions are there for polymerases. Topoisomerases means this is another class of enzymes that is reducing the strain that happens in DNA duplex. Means when that strain happens during the time of, I will show you that. Where during the time of DNA replication, replication is going on. As this replication going on, need to unwind the DNA from this direction. As this unwinding goes on, there's more chance to get develop overstrain in this portion. So it become overtwined here. So and that time, for relieving that tension or strain in this portion, and that time this topoisomerase came and make a cleave here, and in that way it dwindle or reduce the strain or tension that develops there. That is the role of topoisomerase. In the picture we can see that as the tension develops here or strain develops here, that an incision is made or cut is made by this topoisomerase enzyme, and that way facilitates the further process without developing more strain. And this gyrase is also another, its function is also somewhat similar to topoisomerases to remove or to reduce the strain in the DNA double helix. Helicases means it's also helping for unbinding the DNA and some other similar type of proteins. They are all comes under type of motor proteins. How they function? They function by associating with the DNA or RNA molecules. And what is the source of their energy, chemical energy? They got their energy from the polymerization reactions of nucleic acid. As the nucleic acids bind one after other, because of that reaction, it got energy. And also by the synthesis of proteins during the time of protein synthesis, during that pathway also they got some chemical energy, or by the way of ATP hydrolysis. By that way also it is getting its own chemical energy for driving whatever function that they need to perform. Next, significance of motor protein. Significance, now it's very clear to us that because of the motor proteins, it is very easy to process the genetic information, and also a cell can synthesize different types of protein molecules. So that's why motor proteins are very important. That's about that protein. Next, we are going to see about transcription factor motif. It is a topic. It is a area somewhat more relating with the transcription factor itself. So while we are discussing about transcription factor, I have told you that transcription factor have two domains. One is a DNA binding domain, and the second one is a regulatory domain, right? And the DNA binding domain, you look here, you can see so many foldings we can see. And before going to this portion, I am just telling you protein have different organization. Primary structure means a single helical. Secondary means folding or combining with the other. This is beta pleat, and alpha helix. Alpha helix in this way. So secondary. This is the primary. This is the secondary. Tertiary means this secondary structure, secondary conformation again folded together. They underwent many other foldings to get this tertiary conformation. So regarding the transcription factor motifs, we are telling something regarding with this tertiary structure. So as a protein, as we are discussing about transcriptional factors here, this transcriptional factors in its tertiary conformation, it is having so many foldings, and that foldings have some specific pattern. Pattern means because of acquiring such specific pattern, it can bind or it can interact strongly with the specific sequence in DNA. Okay, that folding pattern is very, very important, and that patterns are called motifs. So, what is transcription factor? Motifs, and these motifs are seen in DNA binding domain of transcription factors. What are these transcription factor motifs? There are certain domains in transcription factor as a result of tertiary folding helps to specifically bind with the specific sequence in DNA. So, what are the features? If we are taking the case of a single domain, we can see some alpha helix segment in that. Because if we are considering the case of one helix, sometimes one motif is formed of a alpha helical segment joined with a beta, beta pleated structure. In this way, some motif can form. Yeah, we can say that in one half it possesses an alpha helix, and the another half it possesses a beta segment or beta helix. And using this portion, it can insert into the major groove. Major groove and minor groove we have already learned. Major groove of DNA. Okay. And also along its margin, usually in the alpha helix, the margin means possesses many basic amino acids. Basic amino acids means positively charged amino acids. As it is positively charged, it can very tightly bond with negatively charged phosphate of DNA. Okay, clear? That's what about this main points regarding the motifs. Okay, so features, coming to features, it contain an alpha helix segment, can insert into the major group of DNA because of many different types of forces like van der Waals force, ionic bonds and hydrogen bonds. Next, different types of motifs. So, in that way, motifs exist. Different types of motifs exist in different type of transcription factors. Which are the different types of motifs? Single finger motifs are there. Helix loop helix motif is there. Leucine zipper motif is there, and HMG box motif is there. So, we are going to learn about each type. Sing finger motif. This is a picture of sing finger motif. And what are the features? Is at its base, the base of that folding, that motif, we can see a zinc ion. And this zinc ion is linked by two cysteine that is found in the beta helix portion. And also in other side, it is linked with the two histones that are also part of an alpha helical segment. That's one feature. And so many such domains is possible to occur in same transcriptional factors. And using this domain, it is inserting into the major groove of DNA. Example for a single finger motif is first single finger motif discovered is transcription factor 3A, and it has 9 this type of fingers in its DNA binding domain. Next, helix loop helix motif. This image is shown here. It is composed of 2 alpha helix segments. It is separated by a loop, and it is produced by a stretch of highly basic amino acids. As it is positively charged, it can specifically bind to specific sequence in DNA. And usually this helix loop helix motif occurs as dimers. Dimers means 2 subunits. And these two subunits are encoded or transcribed or developed by uh, two different genes. Next. What is the significance of this one? This transcriptional factor, it is involved in the differentiation of muscle cell, then cell proliferation in the development of certain cancers. How it is found in the development of certain cancers? Goes through gene translocation, means some portion of one gene breaks up and then rejoin with another part or another location in along the genome. In that way, it results in the development of certain cancers. So, this transcription factor, that means helix loop helix, is involved in the development of certain cancers. Not only cancers, it is involved in the differentiation of muscle cells, cell proliferation. Okay. Next, loosen zipper motif is another type of motif. What is the feature of this loosen zipper motif? In this loosen zipper motif, along the alpha helix, every seventh position is occupied by a leucine amino acids. And here two alpha helix are present and it can zip together. So that's why it got the name zipper, and to form a coiled coil. And in that way it exists in the form of a dimer. And the basic amino acids on one side of the leucine containing alpha helix bind with the DNA sequence specifically. Then HMG box motif. HMG box motif's picture is shown here. Reason why it got that name, because it is discovered for the first time in a class of proteins. They're called high mobility group proteins. That's why it is known so HMG, high mobility group of proteins. What are the features? It consists of three alpha helices, and it organized in the form of a boomerang shape. Boomerang, we know its shapes, arc-shaped structure. Example for HMG box motif carrying transcription factor is SRY. SRY, it plays a key role in sexual differentiation in human males. If this transcription factor is absent, then the male develop with the features of female. Next is biological role of nucleotides and nucleic acids. We know that both are very important and they are very related terms. Without nucleotides, there is no nucleic acids. Actually, the polymerized form of nucleotides is nucleic acid. So, biological role of nucleotides. Lack of nucleotide in the diet can suppress the immune system of the body. What is nucleotide? Before that, I want to know that whether you remember what is nucleotide exactly. It is a polymer of phosphate, sugar, and a nitrogenous base. Okay. So, lack of nucleotide diet will suppress immune system, and a nucleotide rich diet can increase the intestinal microflora. Intestinal microflora thrive well when we have a nucleotide rich diet. And the third one, the nucleotide rich diet can accelerate the intestine development and the repair. Then nucleotides are the precursors of DNA and RNA. Next, this nucleotides acts as activated intermediates. Intermediates in many biosynthesis pathway. For example, for such nucleotides are UDP glucose, glycogen, then CDP diacylglycerol. So many are there. Then next one, this nucleotides nucleoside triphosphate, that is ATP, it acts as the universal energy currency for almost all biological processes. Then adenine nucleotides. Adenine nucleotides are components of many coenzymes like NADP, FAD, CoA. These are all coenzymes which have this adenine as its component parts. Then as metabolic regulator, CAMP, we know that in hormonal action, cell signaling, in that pathway CAMP we have learned so many times. So CAMP is as a nucleotide. It has it stands as a metabolic regulator. Next one, ATP dependent protein phosphorylation means certain enzymes are there. If it is phosphorylated, because that means if a phosphate group is added to that enzyme, it become activated. But in the case of glycogen synthase, this is another enzyme. To this, if a phosphate group is added, it is inactivated. By that phosphorylation mediated by ATP, we know it's a nucleotide. That ATP mediated phosphorylation. It is very important in regulation, metabolic regulations. Some enzymes get activated upon addition of phosphate group, and some enzymes like glycogen synthase get deactivated upon addition of phosphate group. That addition is occurring with the help of ATP. Then adenylation of a tire amino acids of bacterial glutamine synthetase. To this enzyme, to the tire amino acid, that adenylation is happening. And what is the effect? Then by that way, that enzyme become the more sensitive to feedback inhibition. That is another regulatory role of nucleotide. And also it can act as an allosteric regulator. Allosteric regulator means in the form of ATP and AMP, because glycogen phosphorylase is another enzyme. As two sides, allosteric site means is not the side for binding with the substrate. To this site, either ATP or AMP bind and activate or inactivate this enzyme. That is an allosteric regulator. Also, this nucleotide functions. These are the biological role of nucleotides. Then biological role of nucleic acids. Biological role of nucleic acids, we know that it is a genetic blueprint, means it's a genetic write-up of a cell, and also it carry instruction for the functioning of the cell. And also this nucleic acids, that means especially RNA is involved in protein synthesis. Is very important because all enzymes are proteins. We know that how it is significant, and also in so many other ways proteins are very significant and inevitable for the existence of life forms. So by this way we finish this chapter in today's section. Thank you.